My name is Caitlin Candell, and this is my Module 4 Cell Bio project. The steroid I chose to do was estradiol. Estradiol synthesis takes place in the ovary, in the mitochondria. It starts off as cholesterol, and through enzymatic conversion in the smooth endoplasmic reticulum, it creates four other steroids: pregnenolone, progesterone, androstenedione, and testosterone. Testosterone then goes through aromatase, which converts testosterone into estradiol. Estradiol then diffuses through the lipid bilayer and enters into the bloodstream. Once it's in the bloodstream, it binds to carrier proteins and acts by binding to nuclear receptors to target tissues that will regulate gene expression and protein synthesis. Estradiol is important because it actually affects the growth and development and plays a role in reproductive functions, specifically in females. The next hormone I chose was follicle stimulating hormone, so staying in that right reproductive path. This starts in the nucleus of the gonadotroph cells, which are located in the anterior pituitary gland. It goes through the rough endoplasmic reticulum, where translation of mRNA happens into pre-hormone synthesis. Through this, it becomes a single peptide, which goes directly into the endoplasmic reticulum, where the protein folds and then forms a disulfide bond. Through glycosylation in the Golgi apparatus, this is then able to be secreted into, put into secretory vesicles, where it will be held until gonadotropin releases the hormone and triggers exocytosis. Exocytosis is a word that just means that it's going to be exiting the cell, where it'll travel into the bloodstream to gonadal cells and bind to the cell surface receptors. This also regulates reproductive functions. And the last thing I looked at was actin. Actin in the nucleus goes from mRNA, where it's transcribed. Translation occurs on free ribosomes in the cytoplasm. As a monomeric actin folds within a chaperone protein and polyenzymes into filamentous F-actin. And this forms part of the cytoskeleton, which we know gives structure to the cell. So actin filaments interact with myosin for muscle contractility and are essential for cell structure, transport, and movement. It does not get secreted. It just acts within.